It is with immense pride and joy that we extend a heartfelt welcome to this August gathering. We are deeply honored to have amidst us the most distinguished Director cum Vice Chancellor of the Institute, Dr. Triveni Dutt sir. We are also privileged to host today's eminent speaker, Director cum CEO of the Fulbright Institute, United Kingdom, Professor Brian Charleston. We also warmly welcome Joint Director Academics, Dr. S.K. Mendiratta sir, Joint Director Research, Dr. S.K. Singh sir, Officer in Charge, Office of the International Relations, Dr. G. Tarusharma ma'am, all the esteemed Joint Directors, HDs, Section in Charges, scientists, distinguished officials from administration, finance, and dear students. We also welcome our scientist colleagues who have joined online, and we gather here at the Swami Vivekanand Auditorium for this special event organized by the Office of International Relations, ICR-IVRI. Today, we are privileged to listen to Professor Brian Charleston's talk on foot and mouth disease virus carriers, virus persistence in individuals and population. To begin with the proceedings, I now request our Honorable Director, sir, to kindly welcome Professor Brian Charleston with a token of respect, a potted plant. Thank you very much, sir. I would now like to request our Joint Director Research, Dr. S.K. Singh, sir, to please welcome our Honorable Director, ICR-IVRI, by presenting him a potted plant. Thank you, sir.
To officially commence the program, we humbly request the esteemed dignitaries on the dais for the traditional lamp lighting, please.
Thank you so much, sir. It is time now to listen to the ICR-IVRI songs, which reflect the values and aspirations guide us in our scientific endeavors. May we have the ICR-IVRI song?
जय जय कृषि परिषद भारत की, सुखद प्रतीक हरित भारत की। कृषि धन पशुधन मानव जीवन, दुग्ध मत्स्य फल यंत्र सुवर्धन। जय जय कृषि परिषद भारत की, सुखद प्रतीक हरित भारत की। कृषि धन पशुधन मानव जीवन, दुग्ध मत्स्य फल यंत्र सुवर्धन। वैज्ञानिक विधि नव तकनीके, पारिस्थितिकी का संरक्षण। शस्य श्यामला छवि भारत की, जय जय कृषि परिषद भारत की। हिम प्रदेश से सागर तट तक, मरुधरती से पूर्वोत्तर तक। हर पथ पर है मित्र कृषक की, शिक्षा शोध प्रसार सकल तक। आशा स्वावलंबित भारत की, जय जय कृषि परिषद भारत की। जय जय कृषि परिषद भारत की। मानव प्रिय कृषि सहचर हितरत, जय हो पशु संस्थान। भारत के कोने कोने तक पहुँचा नव विज्ञान। 19वीं सदी प्रथमोदय, पूना मुक्तेश्वर नव आलय। इज्जत नगर बना मुख्यालय, अभिनव अनुसंधान। बना विश्वविद्यालय, शिक्षण शोध चिकित्सा पशु संवर्धन। ग्राम अभ्युदय श्वेत क्रांति, सहयोगी शुभ वरदान। जय हो पशु संस्थान। विविध व्याधि के टीके निर्मित, रोग अनेक हुए उन्मूलित। वृंदावनी सदृश गौ विकसित, जन पोषक अभियान। मानव प्रिय कृषि सहचर हितरत, जय हो पशु संस्थान।
We now cordially invite the respected Joint Director Research, Dr. S.K. Singh sir, to deliver the welcome address. Sir, please.
Thank you, Dr. Saima and Dr. Anju. A very good morning to all of you. Most respected, the speaker of the today's 136th Foundation Day lecture, Professor Brian Charleston, Director and CEO of the very prestigious institute, Parabright, UK. Most respected Director ICR IVRI, Dr. Trivindra sir. My Joint Director colleague, Dr. Mehndiratta sir, is Joint Director Academic. Dr. G. Taru Sharma Madam, in charge International Affairs. Dr. Roopsi Madam, Joint Director Extension Education. Dr. YPS Malik Saab, Joint Director Mukteshwar. Dr. Sohni Day, Joint Director Cadret. All heads of division, scientists in charges of the various sections, dear students and ladies and gentlemen. Those who are present in this August hall or those who have joined online, I welcome you all. At the outset, I congratulate you all on the 136th Foundation Day of IVRI, very prestigious institute in the field of veterinary animal science, which has a very global repute. It is a distinct honor and privilege to welcome you all on the 136th Foundation Day lecture ceremony of this institute. On this special occasion, we celebrate not only the rich legacy and remarkable achievement spanning over a century, but also our unwavering commitment for advancing excellence in research, innovation, and collaboration in our field of veterinary and animal sciences. We are especially privileged to have with us today Professor Brian Charleston to deliver Foundation Day lecture, whose distinguished leadership at the Purbright Institute has been instrumental in driving cutting-edge scientific breakthrough. Your presence here symbolizes the strong, enduring partnership between two great institutions and fueling shared goals and mutual progress in animal health and disease research. I welcome you, sir. I welcome our worthy director, Dr. Trivindra sir, under whose leadership this institute progress leaps and bounds, and he is the main architect behind this ceremony also, sir. Welcome, sir. Most welcome. I also welcome my all joint director colleagues, head of division, in charges, scientists, and dear students for gracing this function. If you see, as far as the institution like Purbright is concerned, we have seen immense changes and exceptional scientific achievement in recent years in this institute, bringing breakthrough in our understanding of the virus and host interaction under the able guidance of Professor Brian. The Purbright Institute, formerly IH, and ICAR IVRI, they share a strong common interest in the control of major transboundary livestock diseases, including FMD, PPR, blue tongue, and other economically important viral infections. Both institutes are global center of excellence in veterinary virology, vaccinology, and epidemiology, working towards improved diagnostics, vaccine development, disease surveillance, and strategies for livestock health and food security. With complementary expertise, Purbright in high containment viral research and IVRI in field-oriented animal health solution, collaboration and knowledge exchange between the two institutions support better disease preparedness, innovative vaccine technologies, and strengthen global animal health system. As far as IVRI is concerned, if in the past year, the ICR IVRI has achieved a remarkable milestone, including breakthrough in foot and mouth disease vaccine efficacy and progress in controlling emerging genetic infections. These successes are a testament to the commitment, expertise, and collaborative spirit of our scientific community. As we commemorate this historic milestone, let us reaffirm our dedication to fostering knowledge, nurturing talent, and addressing the challenges ahead with renewed vigor and unity. Together, we will continue to build upon our foundation of excellence for many decades to come. Thank you all for being part of this memorable celebration. Thank you very much.
Thank you very much, sir, for your warm words of welcome. To highlight the significance of international collaborations and partnerships, may I now invite Dr. G. Taru Sharma, ma'am, Officer in Charge, Office of International Relationships, to address the gathering.
Thank you very much, Anju. Good morning, everyone, all the dais of the dais. Special greetings to the chief of this institute, Dr. Trivindra, and of course, the speaker of the day. At the outset, I'd like to join my colleague, Dr. Sanjay, conveying the greetings of the day. A decade and 36 years back, when at Pune, we started on this very day at the institute, and it was felt that time that there's a need when we really address all the veterinary-borne diseases. Then came a year when a medical biologist is taken over and has moved stuff to Mukteshwar, and tomorrow he'll be going over there. So I'll rest my voice here for further details. I'll just put it to highlight what all we can do from the Office of International Relations. This is the second lecture in the series as we planned. And when we approached Professor Brian, he was traveling a particular, as a Cape Town last week and so many other things. But he was genuine enough to say, yes, I can make it for 2 days, and that's a happiness, and he's right here. Working with Professor Brian, here at IVRI is our new partnership. We have been working with the Bright Institute, a bunch of our colleagues, especially at FMDB and other related things. And phase 2 is going to be there. So we continue with that. In person, while I was the director in IAB, we have been working and we generated a lot of FCDO fundings, and then came FADH, and his team and our team, we visited each other back and forth. And last 10 years being the chief of Fulbright Institute, as mentioned earlier, in Institute of Animal Health, located at Surrey in United Kingdom, he has really transformed that institution. It's really mesmerizing to see all the glass walls, BSL 3, BSL 4 within that, and always kind of focused, talking, and very big hunch to talk about the viral loads, bacterial diseases, how to address. And I'm so happy, very aptly you chose the FMD carriers, so individuals and population. So I think we all look forward for your talk. Thank you very much for being here. Once again, immensely happy to see everybody over here to witness this foundation. The foundation days are indeed very important to one and all. With that, we look forward. Thank you very much.
Thank you, Madam, for an insightful address on IVRI journey, Dr. Brian's achievements, and the importance of international linkages for advance in animal science research. Now, on this momentous 136th Foundation Day, it's my privilege to invite Honorable Director IVRI, Dr. Triveni Dutt, sir, to share his words of vision with us. Sir, please.
Yes, sir.
A very good morning to all of you. A very good morning to all of you. The Honorable Speaker of today's very important and prestigious Foundation Day lecture, Professor Brian Charleston, Director and CEO of the Fulbright Institute, United Kingdom. Colleague, Joint Director Research, Dr. S.K. Singh, Joint Director cum Dean, IVRI Deemed University, Dr. S.K. Meridatta, Dr. G. Taru Sharma, in charge of Office of International Collaboration, Joint Director colleague Dr. Roopsi Tiwari, Dr. Bhai P.S. Malik, Dr. Sohini Day, Joint Director colleague joined online Dr. Pallab Chaudhary, and head of our valued research stations, the ERS Vilgachia, Dr. Sen, Dr. Gorakmal with his team, he has joined from Palampur, Dr. Vilegaonkar in charge Pune, distinguished head of divisions, my dear scientists and students. First of all, my heartfelt gratitude to Professor Brian, who has accepted our request to deliver the foundation lecture on very important 136 years old heritage institutions, IVRI. And on behalf of IVRI faculty and student, my heartfelt thanks to Dr. Brian, who really came here after a long journey, and I also thanks him that he also acceded our request to visit our mother campus, IVRI Mukteshwar. So, dear colleague, it is a matter of great pleasure that we have successfully completed 35 years, and we are here to celebrate 136 Foundation Day of IVRI, which is really a heritage institute of veterinary and animal sciences. So here I would like to just brief that what we have achieved, what during this our 136 year of glorious journey. So in 10 minutes, I would like to brief that what we have achieved the milestones in terms of teaching, research and education and the outreach activities. So as we are aware that the institute is working with revised mandate since 2023. We have revised our mandate as per the demand of the government and the stakeholder. And we are working with the major mandate with the research, with the basic research, basic strategic and translational research on animal health, production, sustainability. We have also aligned our teaching and student research activity with National Education Policy 2020. As you are aware, this is the policy, strategic reforms implemented by Government of India. And we are also working hard for reaching out to the unreached. We are trying to reach to all our stakeholders. We are also maintaining 14 important, precious germ plasma of the country, comprising of 2,500 animals across of various regions, stations and campuses. The training and capacity building is also our a prime mandate. And basic attribute of the national education policy is to integrate our veterinary education with the vocational education. So under this, we are also trying hard to integrate our all veterinary educations with the vocational education by planning training and capacity building to various kind of stakeholders. And diagnostic is our priority. We are providing regular diagnostic services, issuing national advisories whenever we attend any kind of outbreak across the country. through our cadet center. And we are proud to tell that we are also providing 24/7 facilities, referral facilities through our veterinary polyclinic. Wildlife care management. As you are aware that in the country we have 300 plus national safaris, zoos, rescue center, which, which are maintaining various kind of wild animals. So we are regularly providing health services to various kind of national parks, zoos, safaris, and rescue sector. And by the Act of Parliament, we have been given the status to have a quality control testing of veterinary biologicals. So this is our regular mandate, a very important mandate of the IVRI. Whenever and wherever we are using any kind of biological in the field, or we are importing any kind of biological to the country, this is the responsibility of IVRI. They have to clear first, then the biological will be allowed to use in the field. So this is the 135 glorious years of IVRI. IVRI have been established with the reason that 90% of our cattle mortality occurred during 1868. And during that period, the Cattle Plague Commission was formed. And with the recommendation of Cattle Plague Commission, the Imperial Bacteriological Laboratories, IVL, at Pune have been established during 1889. Then after keeping in view of the requirement and having our natural biosecurity areas, region, this IVL have been shifted to Mukteshwar during 1889. And this campus was established in 1913. So this is our the very impactful journey, and we were into the trainings way back from 1900. We were imparting training to our various kind of stakeholders like veterinarians working in various states and veterinarians working in RBC. So this was the first training programs we have started from 1900. And postgraduate of animal science, postgraduate college of animal science was established 1958. And during 1983, IVRI have been given the status of deemed university, deemed to be the university. And since 1983, we are imparting education in almost all disciplines of veterinary and animal sciences. So to recognize the institution's contributions, Government of India also issued a postal cover and stamp on 9th December 2014. On the day where we have completed, 100 years at IVRI. So this was our glorious journey, and we are providing services to the nation through our 3 important campuses and 3 regional stations across the country, located across the country. We are having a pan-India presence, and it is very pertinent to tell to our young colleagues that our 4 valued campuses, regional stations and divisions have been elevated to the independent institutions. This itself reflected that this institute is playing a greater role in human resource development and the institutional development. So our four important campuses, regional centers, and divisions have been elevated to independent institutions. These are our CIARI Port Blair, CIRI Izzatnagar, CIRG Makhdoom, and Nisad Bhopal. These were our campuses and regional centers previously aligned with the IVRI. So, as for the establishment timeline of IVRI, pre-independent and post-independent. Pre-independence, roughly a total number of four important sections and divisions have been established with as per the demand of the country at that point of time. And subsequently, we have also responded to the demand of the country and not only the country but nearby country. We have also established 40 various sections and divisions at IVRI after 1947. So presently we are having a full-fledged, well-resourced with full of infrastructure, 20 divisions, and we are imparting educations in 20 divisions. So this is in brief our functional diagram that the director and vice chancellor is the principal executive officer of IVRI deemed university, and he has been supported by our 6 joint director colleagues and chief of administrations and chief of finance also. And we have 20 research division, as I stated earlier, 3 campuses, 3 stations, and currently we have 28 supported sections, including 11 scientific and 17 service sections, and 6 well-established livestock farms, wherein we are maintaining 14 important germ plague. The human resource, currently we have 910 human resource working at IVRI, including 201 scientists, and also 709 administrative, technical, and our supporting staff. And these whole our human resource have also been supported by 750 staff engaged on contractual basis. So we have a large number of human resource working at IVRI, roughly 1600 plus. So this is showing our significant contributions. IVRI has made a significant contributions to animal health, including successful eradication of 4 major animal diseases. These are our rinderpest, dourine, CVPP, and African horse sickness. Currently, what responsibility have been assigned to IVRI? Currently, we are also actively involved in controlling of 4 major diseases in the country. In fact, we are providing technical backstopping in terms of giving technical guidance, training programs, and also the required biological reagents to the National Animal Disease Control Program. So this is the flagship programs initiated by Government of India, and our all vaccines used in these our 4 National Animal Disease Control Program have been developed by the scientists of IVRI. So these are the FMD, brucellosis, PPR, and classical swine fever. Our Mukteswar campus, mother campus, and very prestigious campus, we love, each and every scientist love Mukteshwar campus. So this campus has been recently given the status of by the World Organization of Animal Health as a referral laboratories network for PPR. So this is, in brief, the achievement of milestones and achievement of IVRI. IVRI is also providing services in addition to our R&D activities and educational and outreach activities. We are also providing services to the nations through various 6 important ministry, and our 5 services are national importance, including the quality testing of vaccines. And in the quality testing of vaccine, we have been involved since 1969, and also the supply of 12 important biologicals, including 2 for human disease diagnosis since 1936. The disease diagnosis is issuing advisories and capacity building is our also a prime mandate. This services we are providing since 1986, and the wildlife management is very crucial our activity, and government depend on IVRI as far as healthcare management, postmortem, advising various requirement related to the nutrition. And recently we have also going to introduce the wildlife healthcare as a full-fledged division. And we have also decided to initiate the master degree programs in wildlife healthcare as per the demand of the stakeholder and the government of India recommendations. So this is our because besides the health R&D and teaching activities, our mandate is also to work on the genetic improvement of livestock. Currently, we are involved in the genetic improvement of 14 important germplasm. And out of these 14 germplasm, 6 important germplasm have been registered, documented recently. And these germplasm are our Landli, Royalkhandi goat, Chaugarga goat, and also the Gurra pig. And here I would like to mention that in our recent breed registration committee meeting, our 2 important breed have also been registered. One is the Brindavani, and second is the various prestigious indigenous germplasm of this region, which we have defined as a Rohilkhandi cattle. So these 2 germplasm we have added in our assets. And in addition to this, these our registration germplasm, we have also maintaining 4 important prestigious animals at IVRI: Sahiwal, Tharpakar, Murrah buffaloes, and the Brindavani. This breeds, these synthetic breeds of crossbred cattle developed by IVRI. So, as far as our research portfolio is concerned, IVRI has commercialized a total number of 47 technologies, including very important 13 vaccines to 163 commercial houses, wherein we have generated 21 crore rupees as a revenue, and our IPR portfolio is also very rich. A total number of 8 IPRs have been registered, and also we have developed 142 ICT tools for capacity building of our student and faculty. In 2024, IVRI implemented 210 research projects, including 86 externally funded, with a total funding of 154 crores. And if we see the publications profile of IVRI, last year we have published a total number of 310 research paper in various reputed journals, and our average publication per scientist is 2.47. And community outreach activities is also a very important activities of IVRI. Fostering social responsibility is a mandate not only of IVRI, but mandate of all the institutions located at India and also the overseas. So outreach activity, we are all scientists and students involved in outreach activity. And during 2024, we have organized a total number of 1,400 outreach activities, wherein we have benefited 1.4 lakhs beneficiaries. We have a very rich research ecosystem at IVRI, but as per the mandate of the council and the country, we have also developed a outstanding startup ecosystem at IVRI. Through our Raftar AVI and also the AVI of ICAR, Agribusiness Incubators of Incubation of ICAR. So, under these two important programs, AVI Raftar Agribusiness Incubation and also the Agri-Business Incubation of ICR, we have created 40 startup. 40 startup are on board of IVRI. They are expanding their business and they have created by the IVRI. Also, we have developed and expanded a total number of 238 entrepreneurs. So now, the human resource development is a very important responsibility of IVRI. Currently, in tune of national education policy, previously we were imparting PhD degree in 17 disciplines. Currently, we have added UG programs, UG in biotech, BTech biotech, and this year we have also successfully introduced BTech dairy technology. Last academic year, we have added in the academic dimension this master degree in anatomy, and also 2 years back we have also introduced MBA degree programs at IVRI deemed university. So currently in total 40 UG and PG academic programs we have implemented at IVRI, and we have successfully integrated our academic activity in tune of National Education Policy 2020. And the important mandate of this university that we have to integrate our veterinary education with the vocational education. So, in line of this important mandate, we have also introduced 90 important courses, including 23 diploma courses and 67 various vocational certificate courses. And a total number of 70 courses recently we have also included in our academic dimension as a value-added courses. So the timeline given to IVRI that by 2025 we have to fully integrate veterinary education with the vocational education. We have already 70-80% work over. We have already integrated, and by 2035 we have to evolve ourselves as a multidisciplinary institution. So we are, whatever the disciplines important for enhancing the quality of veterinary education, we are already introduced some disciplines like master in bioinformatics, and we have also introduced the master in statistics recently. And we are also planning to include some of the important courses related to big data analytics, artificial intelligence, and other this frontier areas of veterinary sciences. So our target that by 2035 our veterinary disciplines will be the veterinary this deemed university will be the full-fledged multidisciplinary university. And here I would like to mention that one other target had been given to us, that as a whole in the country, the gross enrollment ratio will be enhanced up to the 50% from the 23.7 during 2018. So in line of this important, we are successfully enhancing the not only the discipline but only seat number also in each and every discipline. We are increasing seat number in master degree and PhD degree. Currently, the student intake is 435, and I think during 2016 it was 225 plus, and now the capacity is 435, and currently we have a student on roll 900 plus. Till now, we have awarded 600 degrees, master and PhD degrees with 100% placement. So this is so keeping in view of our quality educations and the qualitative research of our student, we have been assessed and accredited by a very empowered and transparent vertical of Higher Education Council of India as A double plus. So A double plus scoring itself reflected the excellence of our education, creating world-class infrastructure, and also the improving the governance. So this we have with the collective support of IVRI joint director colleagues, faculty, and head of divisions, and also the student, we have attained the highest scoring of NAC A double plus accreditation. And here I would like to mention that this NAC procedure is so robust and strong and aligned with the global best practices. So we have been benchmarked with the global attributes. So we are proud to have A double plus at Indian Veterinary Research Institute, deemed university. So with this brief about the IVRI, in the last, I am extremely grateful to Professor Brand, sparing his valuable time and providing the valuable thought to our IVRI faculty and student. We are really grateful to our all joint director colleagues and in charges of all our regional centers, our valued head of divisions and beautiful student. Thank you very much.
I also thank you to the director. It's been a century, 3 decades, and 5 years. I said 36. So we're starting. Please excuse me.
Thank you, sir, for sharing our achievements and your vision and inspiring all of us to achieve all the goals. And it is now my immense pleasure to introduce you to our eminent speaker, Professor Brian Charleston, whose pioneering work in the field of animal health and virology has been globally recognized. Professor Brian Charleston is an internationally recognized veterinary scientist and immunologist, a fellow of the Royal Society, currently serving as director cum CEO of the Purbright Institute, Surrey, UK. He obtained his BVM from the Royal Veterinary College, University of London in 1982, followed by a master's in molecular biology at University College London in 1988 and a PhD in immunology as a Wellcome Trust scholar in 1991. His early career includes postdoctoral research at the Royal Veterinary College before joining the Purbright Institute in 1994. Professor Charleston's research has focused on the immune response to viral infection in cattle, particularly foot and mouth disease virus, leading to pioneering work in vaccine development. He demonstrated the epidemiological role of African buffaloes in maintaining foot and mouth disease virus in wild populations, a landmark contribution to ecology of this disease. He has led the design of novel virus-like particle vaccine for FMD, which are thermostable and can be produced in low biosecurity facilities, and this can address the global vaccine shortage. His work has significantly advanced the development of vaccines for livestock and zoonotic disease with a strong commitment to improving global vaccine access. Under his leadership, Purbright Institute established the Center for Veterinary Vaccine Innovation and Manufacturing, strengthening global capacity for disease control. Professor Charleston's scientific impact is reflected in over 7,999 citations, an H index of 51, and an I-10 index of 133. In recognition of his outstanding contributions to veterinary science, immunology, and global animal health, Dr. Brian Charleston is honored as a leader whose work continues to safeguard agriculture and society worldwide, and we eagerly look forward to his talk. Sir, the stage is all yours.
To the same pointer. Is that up there or?
Yeah.
Can I get on? Sorry, can I get it on the screen?
Yes.
That's
Both.
How do I do that?
You can use also in this.
Okay.
So you can see
use
it.
Just okay. Good morning, everyone. Thank you for the kind introduction. And I was particularly pleased to hear the introductory talks from my colleagues there. it really reflects. It is a great honor to be here with you. I think it's clear from what we've been told and what we already know of the success of IVRI over the last 136 years. It's been truly fantastic. I had the very good opportunity to have a very brief time looking around the campus yesterday afternoon, and there were a couple of thoughts that came to me. I've been working with IVRI colleagues for nearly 30 years, and this is really on virology and virus host interactions. And I was really struck by the breadth of the breadth of disciplines that are carried out here at IVRI and the other campuses. It's truly fantastic. The going to the farm and seeing those breeds of cattle and buffalo, and the advances being made in the genetics of those animals. Looking at feed, the training of staff, I think, is really important for the next generation of scientists to come through, and I think it's to reflect on 136 years. But I was really struck. This is really a forward-looking institute. It's really looking to how it's going to have an impact on the improvement of animal health and address those major topics that are a concern to everyone around the world. So food security, climate change, and antimicrobial resistance. And I can see that IVRI over the coming years will certainly help address some of those major global concerns. So congratulations to you all. I think this is a fantastic organization, and I'm delighted I've come here to share some of the work we've been doing, and hopefully that will foster more collaborations in the future. I would really look forward to that. So this is a scientific presentation. It's go into a little bit of detail, particularly around immunology. I'll try not to spend too much time going into detail and keep this accessible to the non-experts, but tell a story we've been trying to build over the last 10 years to try and understand virus persistence, particularly of foot and mouth disease. So this slide doesn't really need any introduction here. It's another slide from our World Reference Laboratory at Pirbright. And the areas of the globe that are in grey or a very light colour are the ones that have foot and mouth disease virus still. As you well know, with the control program making great advances in India, but still this is a huge global disease, probably one of the major diseases that needs to be brought under control to improve the efficiency of livestock production. I always like a quote from an economist I know, Jonathan Rushton. He says, no agricultural system can coexist with foot and mouth disease. And I think that's right. All the advances in genetic improvement, etc., are all to some extent negated if you don't bring diseases like foot and mouth disease under control. And the international community have done this before. The Rinderpest is a major achievement for everyone around who was involved in that control program. And we've heard the involvement of IVRI and Mukteshwar to do that. And I think foot and mouth disease is going to be our next big challenge, but it's a much bigger challenge because of the multiple serotypes, multiple strains. But I'm not going to discuss that so much today. What I'm going to talk about is disease. So a little bit of the background science here. So this is on the left-hand side here is the crystal structure of foot and mouth disease, solved in the late 1980s. One of the first structures of this size to be solved. Some really clever mathematics actually to build this structure from the diffraction data. And so just to try and keep the messages quite simple, really. So what I'm trying to show here is that structure on the left, that whole structure of the virus. You can drive antibody production against that structure without using T cells. You don't need T cells to do that. The antibodies are rapidly reduced to that structure. We've proved that. We've proved that in knockout mice that genetically do not have CD4 T cells. And we've proved that by very large quantities, this is all published, very large quantities of monoclonal antibodies to cattle so that we remove all of their CD4 T cells, then we infect them with FMDV or vaccinate them, and you still get an antibody response. But it's not absolute. So on the right-hand side, you've got a high magnification view of the so-called GH loop. These are the structures on the outside of the capsid of the virus, and this is where the virus binds to its receptor, the integrin. So this is how the virus enters the cell. And interestingly, antibodies against that structure do need a T cell response. If in those experiments told you in mice where there are no T cells or where we remove them in cattle, you do not get an antibody against that GH loop. And so you don't get antibodies that effectively stop infection. So it's this mix. You need a mix of T independent antibodies, and you need some T dependent antibodies. I'll go through that a little bit more as we go through the talk. So really what we set out about 10 years ago to try and understand in more detail, actually it's more than that, it's probably 15 years ago. This is how long science takes, as we all know. So there's a lot of talk about the FMDV carriers. This is the persistently infected animals. So it's FMD, as you know, is a very acute infection. Animals get infected very quickly. There's viremia for less than a week, and then high neutralizing antibody. But in the back of the throat of animals, cattle in particular, well, not in particular, but cattle is where it's a major concern in terms of control. There's low levels of virus, live virus detected in the back of the throat of these animals. So where is that virus, and why has foot and mouth disease evolved to have this long-term persistent infection? Why would it do that? It infects an animal, it spreads, it goes on to the next animal. So why does it want to have this carrier state? And there must be a reason behind that. And the other linked thing we wanted to understand is with the current foot and mouth disease vaccine, the duration of immunity is quite short. It's barely a year compared to some other vaccines, like PPR, Rinderpest, etc. We get very long duration immunity, so it's quite a short acting. And so when I was describing earlier these antibody responses that don't need a T cell response, the T independent responses, they work really quickly, but the problem with them, they don't last very long. So it makes sense that if you vaccinate an animal with that structure I showed you, you'll get a very rapid antibody response, but it won't last very long because you haven't driven an effective T cell response. But the duration of immunity after infection is long, yet the structure is the same. The structure of the antibody, the structure of the virus, and the structure of the vaccine is the same. Why have we got that long duration of immunity after infection? I think we may have found an answer to that. And this is a real practical problem because animals become persistently infected even after vaccination, if you get complete stopping of the virus replication. Now, this is maybe not so important when you've got a widespread endemic infection, but as you start to eradicate the disease and control the disease, these animals carrying live virus in the back of their throat become more important. And certainly, for example, in 2001, when we had the huge outbreak of foot and mouth disease in the UK, and also in the recent outbreaks in Germany and Hungary and Slovakia, where they want to return to freedom, they have to go and find those persistently infected animals and remove them. So this is important in the latter stages of a control program. So I'm going to go back a number of years to, I think it was the mid-80s, mid-90s, sorry. So this is a study carried out at Purbeck. It's a little tricky to see the faint coloured lines, but you can see the dark black line there. This is animals. They were infected with foot and mouth disease. And as I said, they became viremic, cleared the virus. And then what we're seeing in the black line is that this is virus isolated from the back of the throat of the animals over time, over about 144 days. So you can see how this carrier there is being measured in detail in these experimental studies. And then eventually the virus won't be detected in the back of the throat. But what this group did is to try and understand what was the mechanism, what was happening with those carriers. They used a technique that's being used for infections like herpes, viruses, etc. So they gave the animals very, very high doses of corticosteroid to try and cause immunosuppression. And they thought that the idea, their hypothesis was that if we suppress the immune system, then you wouldn't have this low level of virus replication. You'd get very high levels of virus replication, and you'd lose control of the infection again. But that's not what happened. Actually, it's quite the opposite, and it confused people for a long time. Let me see if I can get this pointer to work. So here, this is this period of time between these two arrows when the high doses of corticosteroid were given. And you probably won't see this at the back, but here's the line here, the light line following the black line. But as soon as they started giving the corticosteroid, the virus shedding stopped. So that was, how's that working? You suppress the immune system, yet you've completely stopped virus infection. So this was a puzzle for a little while. So I was asked to get involved after the 2001 outbreak. Actually, I was working on immune responses to various cattle viral diseases, and but not foot and mouth disease. But after 2001, I was asked to go to Pirbright and start working on foot and mouth disease and try and understand in more detail the immune response, to try and understand persistent infections and try and develop new vaccines, try and understand how we could develop new vaccines. And I'd already been working on a cell in the lymph node, which I'll introduce in the next slides. And this is some work I'd looked at from the 1970s that, let me skip on from this now a bit more quickly. So what that was showing from that old publication in the 1970s, very high doses of steroid. One other effect that they would cause is a disruption of the structure of the lymph nodes. So this is a lymph node, in the corner of your neck or whatever. When you have a severe infection, you can feel them swollen up and responding to the virus infection, and some really important cells. So this is an EM image of these cells that I'll talk about a little bit during the talk. Is follicular dendritic cells. These are very range cells, don't replicate very quickly at all. In fact, really slowly. If you irradiate a mouse, these cells survive, so they're radio resistant, and they look like an octopus. Really. There's this cell body here with the nucleus and cytoplasm. Then there's extremely long strands here, these dendrites, hence its name. And hopefully you can see on the slide that there's these little blebs here. They're called ichthyosomes, and this is where if you get infected or you get vaccinated, either the virus, for example, will get held on these long strands, or vaccine antigen get held on these long strands. And these follicular dendritic cells form the heart of these germinal centers. So again, this is this lymph node, and if you're vaccinated or infected, the vaccine or virus comes through here and will start a reaction in the lymph node. Now, in this area here outside of the lymph node, outside of the germinal center, there's these short-lived cells. Now, they will produce antibody without T cells. They'll produce antibody really quickly, and they don't need T cells. But they don't produce high quality antibodies. And the reason we think that we've evolved, animals and humans have evolved this really rapid way of producing antibodies, is that so for diseases like foot and mouth disease that replicate really quickly and kill cells really quickly, if you didn't control that infection very fast within a couple of days, the animal would probably die. It'd probably be a lethal infection. And so the body's evolved to produce these very short-acting antibodies outside of these germinal centers. These germinal centers, however, you do need T cells. They contain these follicular dendritic cells that hold the virus or the antigen on their surface. And these structures evolve, grow over 7 to 14 days after the infection. It takes time, too slow for a very rapid infection to bring it under control. But this is where you produce your really high quality antibodies. And so they're essential for development of long lasting, really strong antibody responses. I'll show you a lot more about these germinals and quite remarkable structures, really. They don't exist, you're vaccinated, and the lymph nodes suddenly produce this whole new structure for about 3 to 4 months, and then they go away again. And each of these structures are specific for particular antigens. So if you looked in a lymph node, there'd be one, say, that's specific for foot and mouth disease, there'd be another one specific for another disease. So they're very specific structures, very important for driving immune response. Well, you'll see that from the back. But so what we wanted, after I saw that study where you gave large dose of steroid and we lost the any infection we could detect in those animals, I wondered whether what was happening was that steroid actually destroys that follicular dendritic cell network, destroys those germinal centers. That's one of the side effects of steroid treatment. And so we had the hypothesis that actually foot and mouth disease was also held on the surface of those big follicular dendritic cells. And it's a little bit, probably we won't do it, but you probably need the lights down a little bit to see this. But we've got green staining here, which is foot and mouth disease. We've got the red staining here, which is those follicular dendritic cells that I showed you, and here's the overlay, a mix of red and green. So this was from cattle lymph nodes about 30 days after the animals had been infected and then recovered from foot and mouth disease. And then we looked at the lymph nodes, and indeed we could see foot and mouth disease present in those germinal centers that I was describing to you. Interesting, we had to look at we had to develop new antibodies to stain the virus in that site, because the virus at this time, the animal has very high levels of neutralizing antibody. And so the antigen that's on the virus that's on the surface of these dendritic cells will be covered in antibody. So we had to find antibodies that could find places to bind the virus that wasn't covered by those other antibodies. So it was quite a challenging piece of work. But anyway, we showed that was the case. The trouble with trying to do some real detailed understanding of how that virus is binding to those follicular dendritic cells and what does it mean? What would it mean if the virus wasn't bound to those cells in terms of immune response and what have you? So we haven't got all those reagents available to us in cattle. So we went into a mouse model. We don't like to go into mouse models usually because we like to look in the natural host, because that tells you the truth. But the mouse model was very valuable for these types of studies. And so what we'd shown is that, again, we've got very specific markers for follicular dendritic cells now. This red marker here now, much more precise than in cattle. And here's foot and mouth disease here, and here's the overlay. So exactly the same as we saw in cattle. In mice, we also get this this localization of the virus in areas of the spleen and lymph node after viremia has gone away. So what we wanted to show was how is the virus binding to these dendritic cells? And here's a cartoon of these cells here, as I showed you in that electron micrograph. This small cell body and these very long dendrites spreading out. And I already said to you, so the virus will be coated in antibody this time. So it'll be immune complexed. And again, a little tricky to see, but immune complex is here. And one of the common receptors that's present on these cells that binds to immune complexes is the complement receptor. Complement receptor 1 and complement receptor 2, both together. Because of some really elegant work carried out Kulik in the States, because HIV, human immunodeficiency virus, also binds to these cells and lasts long term for many years actually in people after they have HIV infection. They generated an antibody, this 4B2. So 4B2 will bind to those complement receptors, and the publication was it would stop HIV binding to follicular dendritic cells. And so we wanted to see whether it would do the same with FMDV. So this is the value of going into the mouse model to really try and understand the mechanism. So we did that. We injected mice intravenously with that monoclonal antibody, and then we infected them with foot and mouth disease virus. So this is the viremia data. So in blue, in all of the graphs I'll show you, blue is animals that have been injected with that antibody that binds to the receptor, and red is where they're injected with a control antibody. And we can see that we get good levels of viremia in both the animals that are given that 4B2 antibody or the control antibody. So we hadn't really affected the viremia. Viremia was increased a little bit in the animals that got the test antibody, but all the animals are viremic. So this is data. This is an example of, these are mice with the control antibody, so we should have no effect on that receptor. And you can probably just see here some green color, which is foot and mouth disease in the spleen of the mice. Here's the staining for the network, as I've shown before, and so we've got this co-localization. So the control antibody did not affect the binding of foot and mouth disease to the follicular dendritic cell network. However, in complete contrast, that's quite nice the way that it went away with the next slide, is that we got no staining when we blocked the complement receptor. And so that was the first evidence that we believe that the foot and mouth disease virus that's coated in antibody binds to the complement receptor on these follicular dendritic cells within the lymph node. Those are example micrographs. This is a PhD student, and you might not be able to see the numbers, but they spent weeks looking down a microscope at those spleen samples from those mice and looked at nearly 8,000 germinal centers and counted counted which ones had virus present in them and which ones didn't. And this is the graph that they showed. So at 7 days post-infection, the animals had no viremia at this stage. There's no virus in the blood, but looking at the spleen, none was detected in the animals that were treated with the antibody that blocked the complement receptor, but there's plenty of foot and mouth disease in the control antibody. And a similar story all the way down. There's a gradual decline in the control treated animals, as you'd expect, but at no point did we get foot and mouth disease in the spleen of these animals. So we're pretty convinced that's proof that the immune complexes of foot and mouth disease bind to this receptor on the follicular dendritic cells. So that the virus is being held on those follicular dendritic cells for long periods of time after infection. I won't go through this. This is just super resolution analysis and showing that by using software that indeed the virus and those cells are co-localized. Just as a double check, we did PCR for the virus on the spleen of those animals, and which is very encouraging that we got a very similar result. So we could detect virus genome in the spleen of the animals given the control antibody. No virus detectable in the spleen of the animals that are treated with the antibody. Again, back to the conclusion that the FMD is binding to that receptor. So what happens to antibody response? How important is it to have foot and mouth disease on the follicular dendritic cells in the lymph nodes, in lymphoid tissue, to drive an antibody response? Now we're getting to some interesting basic science, but also interesting in terms of thinking about vaccine design. Think about the importance of what arms of the immune response you need to stimulate to drive an effective neutralizing antibody response to foot and mouth disease. So remember I said earlier, there's rapid antibody response outside of the germinal centers that, but you don't need a T cell response for. So at 7 days, when there's no foot and mouth disease in the germinal centers, there's very little effect on the antibody response. The controls and the test antibody group are virtually the same. But as you move on through the weeks, 14 days, 21 days, 28 days, you can see this is the level of neutralizing antibody. It doesn't increase over time. There's no foot and mouth disease in the germinal center, and so there's no increase in neutralizing antibody response. And here's the titers. Here's the titers from the group where we use control antibody. So that's telling us it's critical that you get either vaccine antigen or virus onto those follicular dendritic cells if you want to drive a protective immune response to foot and mouth disease, and if you want it to be long lasting. So then we looked at total antibody response. So we were expecting also to see an effect on that. And this was one of those experiments that actually quite surprised us. So if we look at IgM here, so back to what I said before, there's these areas outside of the germinal center that don't need T cells, and so you get this rapid induction of particularly IgM antibodies. So that looks pretty equivalent, that not having FMD persisting in the lymph nodes is not affecting that early protective response. But also, we were expecting the levels of antibody in the treated group to be lower than the control group. That wasn't the case. There's no statistical difference between total antibody produced in those mice, but a very big difference in the neutralizing antibody levels that are produced. So that needed us to carry out some more investigations to understand why. A little bit of a busy slide, but let's work on this side first. I talked to you about the GH loop. Really important you raise antibodies against the GH loop because that's how it binds to its receptor, the integrin. The neutralizing antibodies are targeting the GH loop. You can target other areas of the virus, and they could be non-neutralizing. So we looked at what is the level of antibodies against the GH loop in those two groups. And again, there was no difference. So we hadn't changed what the antibodies were binding to, the targets of the antibody. We hadn't changed that. But this was some really, really elegant work that the student did. And they used a surface plasmon resonance. So this is trying to get a physical measure of how strongly those antibodies bind to the virus. And here we saw, you can't see the blue dots here because they're at the bottom. There's very low levels. So what it's saying is, if you don't have the virus on those follicular dendritic cells being held for a long period of time, you produce antibody, but the antibody only binds to the virus weakly and is only weakly neutralizing. Again, reinforcing the point that to have long-lasting, strong protection, you need to have that virus or antigen last for a long time, and being present in those lymph nodes. And that makes sense from understanding why many viruses, many antigens bind to these follicular dendritic cells and last for a long time. They sit there like a library for the immune system. The B cells come in and they bind to the virus that's held there for a long period of time. And the ones that don't bind strongly die. They don't get a signal to keep them alive. So over time, you'll keep selecting for for cells that produce higher and higher avidity antibodies. And so that makes sense. If we've removed that source of antigen for those B cells to keep going back to learn how to produce better antibodies, that's the result you get. You get very low avidity binding antibodies produced. So this also, one of the questions we asked ourselves in the beginning was, what's the difference between vaccine and virus infection? Vaccine, you get a short duration immune, shorter, probably less than a year. Animals are probably immune for most of their life after infection. And one of the major things is that you're getting persistence of the antigen for a period of time, significant period of time after infection in the lymph nodes, and you're allowing that immune response to mature. So again, it's giving us an indication of how would we make a better vaccine? How would we make it last longer? Finding ways to target the vaccine antigen to these cells could be a way of doing that. And with some other antigens, there have been success with that, of tagging antigens so that they do bind to some of these receptors on the surface of these cells. So for us, a really interesting piece of biology, but also thinking about what does that knowledge tell us that can make a really practical impact on vaccine design. So our other question was, why does the virus become persistent? And so there are various reports in cattle and in buffalo, water buffalo, of animals having virus at the back of their throat for a period of time, probably never a year, probably half a year after acute infection. But there are many reports of persistence in African buffalo, not experimental studies, but anecdotal reports from field studies that in fact African buffalo, the virus, it's thought that the African buffalo is the natural host of foot and mouth disease. If you infect a buffalo, African buffalo with foot and mouth disease, it gets viremia, etc., but shows no clinical signs. It's perfectly adapted to that virus in that it doesn't show any clinical signs. But it also, as I say, there are field reports of the virus persisting for many years after acute infection. So we thought, and many people, like including us, have tried to see whether a persistently infected cow will transmit. No one's ever demonstrated it, but the levels of virus in the back of the throat and the duration are quite short, and it may be that just there isn't enough virus. But again, there were field reports of anecdotal reports that African buffalo could indeed infect other buffalo or indeed cattle. So we thought by giving ourselves the best chance of understanding why an animal becomes persistent, we'd better start looking in African buffalo. Now that brings a whole bunch of other challenges because these are people in Africa tell us they're the most dangerous animal there is. They are very, very aggressive. So we're in a situation in that we had some good colleagues in the Kruger National Park in South Africa. There's about 300,000 buffalo in there. They'd done some serosurveys of these buffalo, and nearly all of them seroconvert to all of the three circulating strains of virus at about a year of age. So when their maternal antibody drops, they become naive, and there's virus circulating these populations all the time. And so they almost all seroconvert around the 1 year of age time. And so we thought, okay, let's see if we can try and show whether the virus also persists in the lymph nodes of buffalo. And so we took a chance. As I said, there's 300,000 buffalo in the park. We spoke to our friends who are in the veterinary services, and we darted six buffalo only at random, and we tried to target animals that were about 18 months, 2 years of age. And the vets were very good. When they shot them from the air and we looked at their teeth, they had indeed got animals of about that sort of age. We looked in the lymph nodes of these animals, and we could, in 3 of the 6, we found foot and mouth disease persisting in those lymph nodes. That was an important piece of work because we'd actually tried to get funding for this project, and people wanted preliminary data. So this was the preliminary data. So after carrying out that pilot study, we then managed to secure significant funding. So there's 3 serotypes of foot and mouth disease circulate in African buffalo. SAT, South African Territories, is the full name of that. SAT 1, 2, and 3. And so what we did is those people who work with foot and mouth disease will recognize this. We inoculated the dermis of the tongue with buffalo. So the buffalo are all infected with 3 different serotypes of foot and mouth disease at the same time. Okay, thank you. Yeah, long-term persistence, well over a year, which is when the experiment stopped with the SAT1, but quite different. So the SAT2 lasted for 200 days, which is significant, and the SAT3 for perhaps a little bit longer. So one, we formally show that buffalo indeed have persistent infection for a very long period of time, much more has ever been recorded in cattle. But also there was this difference between the serotypes in the persistence when you gave them together. This is just showing how we looked at we used laser microdissection of samples from the lymph nodes of the buffalo and showed that indeed the virus was present in the germinal centers using real-time PCR and also present in the epithelia close to the germinal centers. So this region here. So the epithelia of the throat that had live virus in was always close to these germinal centers. That will be important later. And this is just to show, I don't need to go into the detail of this. Let me go back. Let's ignore this slide. But what we showed by deep sequencing the virus from this site and this site, so looking at the whole virus, a sort of metagenomic population analysis of this laser dissected tissue, this population was the same as this population. So there must be interconnected in some way, because when you look deeply at the genetic variation in the virus, it was the same in these sites. That's what that slide showing is published if you want to look at it. And some of our colleagues at Plum Island in the States actually published some work as well, which we really found encouraging, is that here's a germinal center, here's some epithelial tissue in the throat. This is actually a cow. You see the little bits of red staining here. This is the virus. And they also had the observation that whenever they were able to stain for virus, it was always near a germinal center. So we believe that that virus that's held for a long period of time is the source of infection for this epithelia, which is then what you detect by swabs or scrapings in the throat when you look for persistent infection. So taking a little step sideways, really, we wanted to understand, we were looking at that difference. Why was the SAT1 lasting so much longer in those buffaloes? Why did it persist so much longer than the other two, SAT2 and SAT3? So we started looking at a cell system, whether we could get a similar sort of competition. So these are let's just look at these two lines here, these two columns here. So this is a goat cell, and this is actually a buffalo cell we created ourselves from buffalo kidneys, African buffalo kidneys. These are the one-step growth curves. Individual infections of those cells with those viruses has very similar growth characteristics. But if you start mixing those viruses together in a single cell preparation, you can see that here again, this in blue, the SAT1 dominates and eventually will dominate the SAT2 and SAT3. So we're getting similar difference between persistence in cell culture that we're seeing in the whole animal. But of course, in cell culture, we can look at a little bit more detail. What is the difference between these viruses? And the difference between this virus that's successful compared to the other two, it enters cells more quickly and kills cells more quickly. So again, that makes a lot of sense in that if this virus gets into cells, kills them more quickly, then there's fewer cells for these to infect. And so over time, this one will dominate just because of this more rapid ability to get into the cell and kill it. So we're quite nice. We've got the individual animal data of buffalo where we saw a difference in persistence. And then we were able to show the same in cell culture and start to understand what the mechanism behind that. So then we did a different sort of study. We were very, very fortunate with this in that within the Kruger National Park, they actually had a 2 kilometer square area behind elephant-proof fencing. I'll show you a picture in a minute, where we could keep a buffalo population in isolation. We kept them in isolation for 4 years to look at the dynamics of the virus infection in those buffalo. So first we did an experimental study. This is gonna take a little bit of walking through, so please stay with me if you can on this one. this is two groups, two replicates. These are the same experiments, but I'll talk through the top one first. So what we did is we got two buffalo, infected them with the SAT1, kept them on their own and kept them for 30 days on their own. We did the same. We infected two buffalo. They became persistent with the SAT2 and the same as SAT3. So we got these two buffalo each, a group of two buffalo, each infected with the SAT1, SAT2, all separated until they were 30 days post-infection. No viremia, but had persistent infection. And this is the colors here for the different serotypes. Then we mixed them with 6 naive animals in the virus and each other. So all got mixed together at 30 days post-infection of these. And this is what happened afterwards when we looked about 60 days after we'd mixed them. And we can see here the SAT1 started here. So these were the SAT1 persistent animals. They were still persistent after 60 days. But it was the SAT1 that infected all the naive ones, almost completely dominated that population, and super infected the SAT2. The SAT3 also did super infect the SAT2. So not only did the SAT1 dominant in cell culture, dominant in an individual animal. When you went into this small population, it was also dominant, and even more so in this second replicate, which is the same experimental design, just a different replica. The SAT1 is again dominating that group of animals. So it just fascinates me. The virus, just by doing a simple thing, found a way to get into a cell more quickly and found a way to kill more quickly, can actually result in it dominating within a population. So this is the element-proof fencing I was describing. This is the 2-kilometer zone that we had about 50 African buffalo in here. They're wild, but great scientists and vets, but they don't know how to park cars, do they? It's chaos, isn't it? But, anyway. So month we'd collect them into this holding pen, and then we'd dart them with dart guns. You can't sample these animals. Then go and sample them when they were asleep, taking tonsil swabs, taking blood samples, etc., looking for were they persistently infected and what were they persistently infected. So this is one figure from the paper I should have. That's 4 years of work looking at these buffalo and also doing some mathematical modeling. So remember, this is about 50 buffalo held in isolation, and so there's no new naive animals coming in other than newborn calves. This is a reproducing herd. There are bulls in there, or bull in there. So this is the data that would predict, the mathematical model would predict for a group of animals infected with either SAT2 or SAT3. That one, as we've seen, goes in, infects really quickly, and then all the animals become immune, and then they stay immune. And so very quickly, if you didn't have persistent infection, the virus would expire. It would become extinct because it has nothing to infect. Infects things and so quickly. The SAT2 infects things, goes through the population slightly more slowly, so will actually last for a couple of months, but still not enough. And then the SAT3 also spreads slightly more slowly with only acute infection, but also would become extinct within a period of time. Now, the reason these will become extinct, and this is really why we think animals become persistently infected, because the buffalo calves seasonally calve about once a year. And what you need to do with your foot and mouth disease, especially in this isolated population, you need to go from one generation to the next. So mum has got to get infected with foot and mouth disease and be persistent. She has a calf. It has maternal immunity, and when it becomes naive, it can then infect that population, that naive population, because it's got persistent virus in the back of it. That's what this model is showing here. So one very acute infection spreads really quickly, but is very good at persisting, and so persists in the population. SAT2 becomes extinct. within that 40-50 buffalo population, and sadly become extinct over the period of that, but it's not as good as that one. Really pleased, predicted from the model, and after that 4-year to bridge that population, to get to the next cohort of naive animals once their maternal immunity's gone away. Right, let's see if this works. You'll have to come and help me run the video, please. So this is what I've been telling you earlier and showed you some images. There's the germinal center. Oh no, can we go back? Sorry. There's the germinal center, and this is the epithelium. And what I told before is that the virus circulation here is the same as here. Now, we know there are mechanisms to get virus from here to here. There's dendritic cells, etc. There's ways of doing that. But if this is the circulation here between how does it get from here to here, we feel like we understand a lot more about foot and mouth disease, but as always, there's always more to learn, and we don't really understand that. Yeah, thanks. So if we can try and run these videos, this is nice work done by Jason's sister groups. In green are B cells, and in red is antigen on the follicular dendritic cells. We'll see it better here. So here's a B cell. It comes in, it actually grabs a bit of that that's on the follicular dendritic cells and migrates away. I always think, well, that's Hennig on there in this experiment. What if that was live foot and mouth disease? B cell comes in, picks up the virus, gets infected, and then is able to spread locally. It'd be nice if we could prove that, but it's a nice idea of how this might work. And so this is my final slide, just to say thank you for listening. These are cattle on the edge of the Kruger Park being vaccinated against foot and mouth disease, just in case the fences come down and buffalo break through to mix with their herd and cause foot and mouth disease infection. Thank you very much.
Thank you, sir, for your wonderful talk on FMT with extraordinary depth on its immunological aspect, especially the dendritic cells. It's like a host pathogen interaction, how the natural infection and vaccination interacts inside the host. And personally being an immunologist also, it's much informative for me also. In our country, still FMT remains an most elusive virus and most devastating pathogen. So your talk will definitely enlighten our scientific fraternity for its effective control also and further research also. So we shall now move to the interaction session. We will have a 15 minutes interaction. So I request the audience to please keep your questions precise and focused. And first we will request our scientists who joined online from our various regional stations. We request our scientists who joined online from the regional campuses and stations to please interact with Professor Brian Charleston.
Hello. Am I audible?
Yes, sir, you're audible.
Yeah. Myself, Saravanan Paramasivam. Sara, I think Professor Brian Charleston may be knowing when I visited Fulbright in 2009. And I worked with him. I had an opportunity to work with him. That time he was working on this lymph node drainage and all this germinal vesicle, all this information on persistent carrier status. It was an excellent talk.
Sir, sorry to sir.
Yes.
It's not that audible, sir.
Am I audible now?
Yeah.
Now come to the question directly because we have a large audience. Please fill up the
Yeah.
Please come to the
It was an excellent talk by Professor Brian Charleston. We are very much thankful to him. And my only we are India is moving forward forward for FMD eradication by 2030 by zoning and also by free zone creating free zone clearance, all this. I request Professor Brian your advice for eradication to achieve quickly, and what are the vaccines we can have it as an emergency stock during the later phases of the eradication program. Thank you.
I think the question was, can we have emergency stocks for foot and mouth disease vaccine?
Yes,
Would that
yes.
Okay. So it was actually in 2005, I was set the challenge by DEFRA. So this is our Ministry of Agriculture. So particularly in free countries, there's a problem in that the vaccines, because the vaccine is so unstable to temperature and pH, the vaccine banks are stored in liquid nitrogen. And so if you want to have an emergency vaccine, you have to thaw the antigen. I mean, we're now talking about large quantities. You then have to formulate it with adjuvant and then bottle it and then distribute it. And so that can probably take, if it went really well and in smaller quantities, a week, but it would probably take you 2 weeks. I always remember when I first started in foot and mouth disease work, somebody who was just retiring then, he said to me, there's one thing you need to remember with foot and mouth disease. Things happen really, really quickly. And he's absolutely right. It really infectious virus. 2 weeks is too long. And so the challenge set to us was, could we produce a vaccine that could be stored at 4 degrees, ready formulated for a long period of time? And that's taken us a long time to do that, particularly for we've got 20 different strains now that cover the serotypes and the different strains within the serotype. But by making a completely synthetic foot and mouth disease vaccine that mimics that capsid structure I showed you in one of the early slides, and then introducing some molecular changes into that capsid so that it's physically stable. So these capsids are now stable at 56 degrees C. And we have some storage data where formulated vaccine can be kept stored intact, ready to use for over a year. And it's only for a year because that's as long as we've measured it. And we've no reason why it shouldn't go on for longer. So that makes a really big change. So if say we had an outbreak, you wouldn't have that decision point. Do I start vaccinating? I won't get the vaccine for 2 weeks. It's too long. No, do I start vaccinating? I can start vaccinating tomorrow. I just need to move the vaccine out there. So we think that's going to make a big difference.
Thank you.
Thank you, sir. So next, any question, please? Sir, can you please leave your question in the chat, please? Sir, please put your question in the chat, please.
From chat.
Yeah, meanwhile. Okay. So you can ask. So please ask also, please.
Yes, sir. Sir, good morning, sir. I am Dr. Tamil Selvan from IVRI Bangalore, sir. So just I want to have one question that, so you have shown that SAT1 is comparatively more giving rise to a career status. So recent days also we have seen that a lot number of SAT1 outbreaks in the world. So how you see that this particular carrier phenomena is also an important factor for the SAT1 spread in the recent times.
Yes, thank you. I think we can learn some things. So to get a better duration of immunity, really. So the vaccines are very good. They induce a very fast immunity, but where they could be better is it induce better long-term immunity. We know the current vaccine only induces a modest or poor T cell response. So finding ways to stimulate a better T cell response, I think would help and make a better antibody response and longer antibody response. And there may be ways of doing that by using T cell epitopes from things like tetanus toxoid or cholera toxoid. They could be attached to the capsid. That has worked with childhood meningitis vaccines, for example, where conjugating a T cell epitope to the carbohydrate of the bacteria has been extremely successful at increasing the duration of immunity. And then is it possible to find ways to target the vaccine antigen to the follicular dendritic cells? And there was some early work using not a real vaccine, but a protein similar to hen egg lysozyme, is by adding a tag to it so that it would bind to the complement receptor, as I showed you for foot and mouth disease. Could you enhance your duration immunity? That's worked with some antigens and hasn't worked with others. And my colleague here next to me was telling me yesterday, reminding us yesterday of the immune complex infectious bursal disease vaccine. How's that working? Immune complexes would sit in follicular dendritic cell network. So I think those two things, either target the antigen to the follicular dendritic cells by some sort of tagging mechanism, or by finding a way to drive a better T cell response.
Thank you, sir. Is there any other question from online?
Yeah, well, it was a very fascinating and a very informative talk. Thank you so much. I'm Arnab Sen speaking from ERS Kolkata. Am I audible?
Yes, sir.
Yeah, so I have got two questions, Professor Brian. The first one would be what is the exact status regarding persistence in pigs? Because some workers have reported that pig alveolar macrophages can carry this virus, but there are other studies which say that pigs can clear the infection in 3 to 4 weeks.
A fascinating question. I should have mentioned that in my talk, and thank you for bringing that up. So the pigs prove the rule that we've shown in cattle, in that pigs, if you infect a cow with foot and mouth disease, they're immune for long periods of time, maybe even life. If you do the same with a pig, that is not the case. It's short duration immunity. And other groups, not us, Spanish group and others, have looked in the lymph nodes of pigs, and you don't get this retention of antigen. So in some way, that's I think it's not proof, but it's good evidence that you do need that persistent to get that long duration of immunity.
Yeah,
Thank you, sir.
thank you so much. Thank you.
You can ask some people from
Yeah. So we can have from our audience offline, please.
So somebody's asked the difference between persistence and carrier. They're the same thing. It's just people use different terms. It's no difference.
Yeah, like thanks, Brian, for such a wonderful insight. My question relates to what was inquired by Dr. Chandrasekhar from Bangalore. I'd like to know which among the three, SAT1, SAT2, SAT3, came first from the evolutionary stage, and what special properties does and what is that special character that SAT1 has acquired over the other two to be persistent, longer duration, and also perhaps multiplying to a higher factor than the other two? Have we understood this?
No, the simple answer is we don't fully understand that yet. But be very clear. So this was the models I showed you were in an isolated population, much like island populations of people. Some infections can become extinct in a closed population. Now, clearly, SAT1, SAT2, and SAT3 are amongst the wider buffalo population where you've got, the herds are not static. New susceptibles are moving in and out. So that's why. And our mathematical modeler, he did some calculations. You'd need a smaller population size to keep SAT1 going if they were free living, and you'd need, tens of thousands of buffalo to keep a SAT2 infection going. So these infections will persist, but it's just giving us an idea of the different fitness of the two viruses or the three viruses. So what we think, and we more work needs to be done to show the exact mechanism, but that there is a strong correlation between the speed of entry of the SAT1 compared to the others into cells, and also as a consequence, well, we don't know this, but if a virus enters more quickly, it's more likely to kill the cell more quickly. It may also have some other mechanisms, such as the proteases, etc., may act more quickly. But we've done this work a little while ago, and as all things, you're writing it up and getting reviewed. But there's another example of the Omicron variant of SARS-CoV-2. So that wasn't more virulent, but was certainly more infectious. And that actually had evolved a different way of entering the cell, a different, quicker way of entering the cell. So there was another example of a virus could enter a cell more quickly and so spread in a population more quickly.
So maybe when we understand this better, we may be able to engineer better vaccine, possibly modulating all the key mechanisms to develop longer immune response.
So yeah, I don't think that bit, understanding that link between speed of infection, speed of killing, and will necessarily help with vaccine development. What I think it will really help us with is understanding the phenotype of virus from its genotype, because again, as you know, we with modern sequencing technologies, we can get sequences of viruses really quickly. But the hard work is if you get a new virus emerges and you see the sequence, it's predicting how bad is that going to be, how well will this spread, what pathology, and I think for me that's the next big challenge of virology is, can we more reliably predict the phenotype of a virus from its genotype, and there. That's incredibly valuable. You start thinking about control campaigns really quickly. If flags come up that this newly emerged virus, this is going to be a really bad one, then I think that's where the real value of this will come.
Thank you.
Thank you, sir.
Thanks for a informative lecture. My concern and my skepticism, since I look after wildlife, in 2019, there's a place called Chhatbid in Punjab of India. We had mortality due to FMD in chinkaras, blackbuck, which is a schedulable animal, then goral, Indian bison. And our hypothesis was that the leftover concentrated mixture was thrown outside, where the stray cattle or feral cattle population grazed over there, it infected these animals. Somehow, as you have said, 2 weeks was a too long a time. But then, this is a free range wild population sharing the same environment where the captives were kept. Now, my curiosity is, can we go for oral vaccine of FMT for these animals? Because capturing them in a country like India, if there is one death during capture, it will be stop of the town. Secondly, we kept mum till 2024. There was no mortality. Recently, in 2025, in September, in a centrally located zoo of Pune, we had 6 mortalities of deer. And somebody diagnosed it as angiotic hemorrhagic disease. Somebody said, no, it's a botulism. Finally, our PDFMD diagnosed it as FMD. And recently in Ranchi, on 2nd of December, there are 6 mortalities again in animals. So my concern is about the vaccination of wild animals. Thank you so much.
So the first question is easy to answer. There's no prospect of an oral vaccination at the moment. The only likelihood of oral vaccination for anything, or I think the best example of intranasal vaccination is the bovine herpes virus vaccine, which is live attenuated. So I think you would need, using current technology, to have a live attenuated vaccine to do that. Nobody is willing to take that risk with foot and mouth disease because we can't get that balance between efficacy and safety between that. So that at the moment is off the table. And the other thing, we don't have the delivery mechanisms, mucosal vaccines to do oral vaccination with killed antigen. That technology is not there yet. Your other question is really interesting, actually. I mean, I wasn't aware of that wildlife situation, but when you were saying that, it just reminded me that so generally foot and mouth disease doesn't cause mortality. Sometimes in young animals, you get the so-called tiger heart, where you get myocarditis, especially in younger animals. And I think where you do get case of mortality, it's quite often a myocarditis type event. Again, getting back to the tropism of the virus has changed or something. But I can think of 2 examples where we've been surprised at the large number of animals killed. This is now domestic animals. I won't be able to answer your question directly, but I see the parallels in that. So there was an outbreak of a SAT virus in Egypt. So this was a virus, a serotype that hadn't been seen in that cattle population before. And that caused quite high mortality, around 10% a number of years ago. And then we saw another outbreak of a new SAT isolate that is thought to have come out of Ethiopia and gone into the Middle East. And that also caused high levels of mortality. So again, that was a surprise to us. And it's back to the previous question, really, is there must be a signature in those viruses that we don't know at the moment that predicts whether that particular virus will kill or not. My guess, as an answer to your question, is that there's something about that host virus interaction that is causing the death of those animals. In that, and I don't know if you did the pathology, but I'd be really interesting to know if that was myocarditis that killed those animals, or another mechanism. But that seems to be the usual way. But if we could try and get, we don't have enough data. That's the trouble. To try and interrogate those sequences, we don't have enough data of non-lethal and lethal foot and mouth disease to attempt to try and determine what the cause of that is. But very interesting. Yeah.
Brian, thanks for a very engrossing talk to given. This is Mother from IVRI, this institute. Very interesting to hear about the persistence of this infection in wild buffaloes. If I can extrapolate that data to the Indian buffalo populations, the persistence of this virus to the young ones. And in this country, we are chasing a dream of getting rid of this virus by 2030 by a very large program by the government on NADC. Are we trying to chase a dream which can be never achieved because of the persistence of this virus, as your data predicts? So, because there's a lot of talk in the field also eradicating the virus. Until we get eradicated of this virus, this program is not good.
No, I don't think so. From my understanding of the literature and talking to so our friends from the States did work in the east of Asia looking at water buffalo, and the simple way of explaining it, the persistence in water buffalo is very similar to cattle. It's not long duration. There isn't that. These are African buffalo, quite a different species, and that persistence, that lack of clinical signs, I think that's very different. I think So no, I think if the Indian authorities carry on in the way they're doing, then I think that it is achievable to achieve foot and mouth disease control and then perhaps ultimately eradication. So I don't think you've got an intrinsic problem with the animals that you have in India. I have a very different conversation with people in Africa. I don't know how they're going to. They won't be able to eradicate the virus from Africa. That would be very, very difficult to get rid of that persistent infection in the buffalo. And so I think you're going to be in a, as they do, there's fencing to keep buffalo away from livestock, or you'll have you'll have free from FMD with vaccination, but you'll constantly have animals being challenged from that wildlife reservoir. But I don't think that's the case in India.
The African buffaloes manifest very obvious clinical disease.
No.
While
Oh.
the Indian water buffaloes do not.
No, so African buffalo don't show any clinical signs. None at all. Yeah.
Can I have one last question, please?
I can't hear. Yeah.
As we explained that in natural just for longer
Still can't
hear.
Sir, please louder, sir.
So is there any relationship with the display of antizin on the FDC and then determination by those B cells which are entering into the hypernutrition and following selection of the receptors?
Yeah, we don't know in that much detail because we can't look at it. But what so I don't know is the answer to that question. But what we've done is what we can do is look at the B cells that come out into the population in the circulation. So post vaccination and post infection, we look in the blood. And we can see memory B cells, and we can see long-lived plasma cells. They migrate from the lymph node. If you set up an efficient germinal center reaction, you'll pick up those cells migrating from the lymph node to the spleen and bone marrow, etc. And we can certainly see those populations. We see a spike of B cells about 3 days post vaccination or infection, and that's shown. Our interpretation of that is that that's showing evidence that germinal center reactions happening, and we can see we can show with a boost. So we get a very little response after primary immunization, and after boost we get a really big burst for about 2 or 3 days of those long-lived plasma cells migrating from the lymph node to the bone marrow. So again, I think that's a good indication that we've driven that germinal center reaction. I think that's the way we'll do it. That seems to be pretty good, actually. I mean, people are doing that in humans and mice and what have you, and the data is certainly looking quite strong.
Like this was last we thought, but there's a few then hope Brian, you don't mind taking the question.
Yeah, no, it's fine.
And then others I request really in the ground. Let's please.
Good morning, sir. My name is Sigrid Ayasam. I'm a student of BSc in English first year and I'm question for you. For the infection studies, would it be wise to use the diffusion equation for the virus in the various different samples of animals to check the rate at which the virus infection proceeds?
I didn't get that.
You're a first year BAC student, right? No?
Yes.
So can you just focus on your question? Just precise. Please repeat once more.
For the infection studies, would it be wise to use the diffusion equation for various tissue samples to check the rate at which the infection proceeds?
He's asking about the diffusion equations to check the different samples for the virus persistence. You are clear about what you have asked?
Yes.
It's okay.
So he's asking, can we use equations?
I still didn't really get it. The diffusion equations for what? So to check between virus samples.
Yes, sir. Like how from the initial receptor binding of the virus to how it enters the cell and how it rapidly spreads throughout the cells.
Means you are asking about the variation in the virus binding to the receptors
No, ma'am, the
Mathematical.
I suppose, come to the ground, you will interact a little more, right? Thank you.
I'll come and speak to you later.
Yeah, something
Come see me.
other than
Yeah.
natural virus pathology. He's asking about the mathematical equations
Yeah,
of
yeah,
the
okay.
diffusion. So I hope we can conclude.
Yes, please.
Yeah. Okay. So we sincerely thank Professor Brian Charleston, sir, for his sincere and patient listening to our science and students. So the depth of the science you share will continue to inspire us in our teaching, research, and in our disease control efforts, sir.
Thank you very much. Well, thank you. I think it was a very fruitful and very enlightening interaction session. And it is time to say thanks to our eminent speaker. And for this, I would like to request our director, IVRI, to please present a memento as a token of our gratitude and appreciation.
Can make something.
Thank you very much, sir.
अरे बाहर वाले को बोलना ही नहीं है उसको भाई
To formally acknowledge all the contributions of today's successful event, I now invite Joint Director Academics, Dr. Mehdi Ratta sir, to propose the formal vote of thanks.
So, most respected Director cum Vice Chancellor Dr. Devinder sir, today's guest Professor Brian, my joint director colleague, joint director research Dr. S.K. Singh, Dr. Taru Sharma, officer in charge, Office of International Affairs, my joint director colleague joined offline and online, all head of divisions, in charges and scientists and dear students. So it is my privilege to offer vote of thanks on the occasion of this Foundation Day lecture. So we are deeply honored to have among us Professor Brian, Director and CEO of Purbright Institute, a world-renowned center of animal virus research and global reference of foot and mouth disease. Sir, we are really thankful to you for accepting our invitation and coming only for this lecture from UK. So thank you very much for accepting our invitation, sir. Professor Brian lecture not only enriched our technical understanding of FMD virus biology, pathogenesis, and immunology, but also shed light on the global efforts needed to control such transboundary livestock disease. His pioneering work on immune response of FMD virus in cattle and also this African buffalo and development of novel vaccine and insight into virus-host interaction exemplify the highest standard of scientific excellence and translational relevance. We are particularly grateful for the generous sharing of Professor Brian's global experience, including how surveillance and vaccine innovation at Purbright contribute to the safeguarding livestock. For us in India, where FMD remain a major animal health and economic challenge, especially related to the duration and stability, so these insights assume great significance. So we are really thankful to Professor Brian for all this. So I would also like to thank our dynamic director for making all the efforts in improving the global linkage of IVRI. I would also like to acknowledge the efforts of Dr. Taru Sharma and all the organizing team and all the hard work they have put to ensure the smooth and fruitful scientific program on this occasion of this Foundation Day. So in the end, I also, again, I want, on behalf of everyone, I offer our warmest thanks and deep appreciation to Professor Brian, and I hope that this marks the beginning of our future collaboration, exchange of knowledge, and strengthening bond between IVRI and the Fulbright for the benefit of livestock, science, and society at large. So thank you once again. Thank you one and all.
Thank you very much, sir. Before we proceed to the end of today's function, I have a small announcement to make. We request you all to reach the IVRI grounds for the validatory function of 136 Foundation Day and Sports immediately after this program. And after the national anthem, I request you all to please assemble for a photograph with Dr. Ryan. So I think first I request you all to rise for the national anthem.
तब शुभ नामे जागे तब शुभ आशीष मांगे पाए सब जय गाता जन गण मंगल गायक जय हे आनंद भाग्य विधाता जय हे जय हे जय हे
Thank you. We'll have the photograph in the hall itself.